Hi everyone, I'm Neja Baga. I'm a computational biologist at the University of Oxford, and today I'm testing Claude Science on a real single-cell RNA sequencing data. I'm not interested in whether it can write R code. Plenty of AI tools can already do that. What I want to know is whether it actually makes my pipeline easier. Before we start, there is one important thing to understand. Claude Science is not a new AI model. It's the same Claude that's already available today. What's new is the environment around it. Instead of jumping between RStudio, the terminal, documentation, notebooks, and different tools, everything happens in one scientific workspace. So the interesting question isn't whether Claude became smarter, it's whether this new way of working actually makes research easier. How it fits together. You give the goal. A coordinating agent runs code locally, reaches literature and databases, and applies domain skills. Everything comes back as an auditable artifact. Your data stays on your machine by default. Persistent session runs code locally. Every figure ships auditable and it scales out when you need it. For demo, I choose PBMC3K datasets, about 2700 immunocells, the hello world of the single cell. I choose it because I already know what the right answer looks like. If it drifts, I see it. Here is the Claude for the Science beta version website. As you can see, no is available for the Mac and Linux. Actually, I'm not about to explain how you will be able to set up Claude for Science on your own machine because if you follow the documentation of the Claude, it's very straightforward. You will be able to figure out. I would like to show you a pipeline from zero to end. Before anything happened, this is the data set. It's just a folder on my laptop containing the original 10x genomic files. Nothing has been uploaded anywhere. And this is the Claude Science. It looks like a browser window, but it's actually running locally on my machine. The language model itself lives in Claude, but the code, the data, and the computational all stay here. When Claude wants to read the database, it asks for permission. Right, let's run the analysis. I'm going to skip over package installation and environment setup, which has a lot of value for a computational biologist. Even I don't have to think about that because all of them is doing by the Claude in the background. And it provide an explanation about the data and the pipeline that it would it want to run on my data set, which I confirm and approve and ask the Claude to go forward and do it. As you can see, there is a step here named setup and environment and run the command. For running each of the command, it asks my permission to do that. The first interesting moment came during the QC. It provided me all the QC plots which I could get from the Seurat pipeline. The first interesting moment was here. Like almost every Seurat workflow, Claude suggests filtering out cells with more than 250 detected genes. This is usually described as a doublet filter. So I asked a very simple question. How many cells did this actually remove? The answer was 5. Just 5 cells is removed by the Claude. But I know that For a dataset this size, I expected something closer to the 60 or 70 doublet. So I asked another question. Where did the rest go? That's when Claude changed its explanation. It points out that this filter mainly cache obvious doublets if two cells are the same type end up together. They often look almost identical to a normal cell, so those homotopic doublets pass straight through. In other words, this filter only removes the most extreme cases. Yeah, I've used this filtering step countless times. It's in almost every single set tutorial. It took one question to expose its limitation, but somebody still had to ask the question. It's provide me the elbow plots and also regarding the PCA, I challenge it about the PCA that is correct and also which of them biologically make more sense, which it accept. And then it provided me the different clusters and as well as the true labeling for each cluster and the gene expression profile of them, which was super interesting to have all of them in one place in one platform and run and set up in the background. Also, it was some explanation which catch this time by the agent reviewer, not me. Which was it was mentioned that NKJ7 is specific to the NK cells, which is not necessarily true. And this is something were catched by the reviewer. Even the clustering was right, the annotation was right, and also the gene expression dot plot was right, but the explanation had minor issue, which the agent reviewer correct that one, which was super interesting and I like it. I also challenged the different decision and each time it was a good collaboration between me and Claude to understand and finalize the final result. And at the end, I asked the Claude to provide me a method section, which is provided to me with a good structure which I like. So, as you can see, every files that during this discussion and also before and after got corrected is saved in the files folder in my project and also all of them is version. There is a notebook here when you see all the code is developed for providing uh, these plots, which is interesting as well. As a wrap up, would I use Claude Science as a computational biologist? Yes, I absolutely. Not because it's replaced me. It doesn't, and it doesn't replace Seurat either. What is replaced is a huge amount of the work around the science, environment setup, software versions, package management, keeping track of scripts, reproducing fill figures months later. All of that becomes dramatically easier. This is still a beta product. I tested it on a benchmark where I already knew the expected answer. That's exactly what a benchmark is for. And this is only one pipeline that I run. And it was interesting that at the end it asked me to save it as a workflow to be a reusable skill. If you go uh, here in the customize, you will be able to see the different skills which already feature and imported in your session, and you will be able to modify them or add the new skills by yourself, which I will explain more in my future video. And yeah, Next time, I want to test it on something much harder, a data set where I don't already know the answer, multi-sample integration maybe, trajectory inference, or something genuinely exploratory. And if you're a computational biologist, I'd love to know where you would have challenged it first. Thanks for watching, and I'll see you in the next video.